Okay, well, welcome everybody. It's an absolute thrill to be here, to be able to talk to you about our favourite topic. So we don't always get the chance to do that to such an interested audience. So we're really looking forward to the day, and we hope that you enjoy our hospitality as well. Okay, so as what I aim to do is to just give you a quick overview of vitamin C, a little bit about what we know and some of the new areas that we are becoming interested in terms of the expanding knowledge about this topic. So just to start, everyone in this room will be aware that we, unlike most of the animal kingdom and all of the plant kingdom, are not able to make our own vitamin C. So for us it's a vitamin. For most plants and animals it's just a thing that their bodies make. And so we, together with an odd mixture of other species, somewhere in our distant past we lost the capacity to make vitamin C. So most animals make it in their liver, a few species make it in the kidney, and so for us, we probably wouldn't be sitting in this room if we all made vitamin C, because it would just be something that regulated itself. So what is it, this thing that we're talking about? So it's made from sugar, as in the previous slide. And where it's interesting is in its ability to undergo redox reactions or oxidative reactions. So you'll be hearing a bit today about the various implications of this. So this is ascorbic acid, which is vitamin C in its complete state. In biological systems and at neutral pH, it exists as ascorbate, so it's an anion. So that makes its transport into cells a little bit difficult because charged molecules don't just diffuse in and out. So this species undergoes 1 or 2 electron oxidations to become dehydroascorbate. Now that can be recycled in your body and regenerated back to ascorbate. So this is the active species. If it is turned to dehydroascorbate, it's quite unstable and will very rapidly break down. So within minutes at neutral pH, it will break down to diketogulonate and other species, and that's irreversible then. So this is how we lose vitamin C eventually, so we don't just take it up and it's there forever. So this oxidation reaction is exacerbated by exposure to oxygen and by heat. And so this is why you destroy your vitamin C when you're cooking your food. And then this product, that's an irreversible reaction. This is important in biology. So it's an excellent reducing agent. It also forms complexes with transition metal ions, particularly iron and copper. And this is important for its role in biology. So what happens when we eat vitamin C? So we take it in through our diet. So we get, pressing the wrong button all the time. We get most of it from fresh fruit and vegetables. We eat it from our gut, it's taken up into our bloodstream. This is really important. The concentration that ends up in the bloodstream is what determines where it goes into the tissues. From there, it disperses into all of the tissues in our body and is actively taken up. And what you'll note is that the concentration, the intracellular concentrations in all our tissues, much higher than it is in the bloodstream. If you're an animal, and that made your own vitamin C, this distribution pattern would be the same. The only difference is that initially the vitamin C would come from the liver and go into the bloodstream, but then the same process of dispersion. So that's the only difference between us and the animal. So it's not made everywhere in the body of an animal. The same process of distribution applies. So if you take in, once your blood concentration gets up above about 100 micromolar, then as it's filtered through the kidney, any excess passes into the urine. So your body works hard to maintain the plasma level, or not to allow it to be above 100 micromolar, certainly for any length of time. Just go back to that one more point. One thing, you'll notice that there's quite a difference between the amounts that's in various tissues. Lots of these numbers are quite historical, and people thought originally, why is that? Why does the brain have really high levels? The pituitary, the corpus luteum, there's some like odd tissues that have really high levels. The thought was there's something, those tissues need vitamin C for a particular purpose. Well, as it turns out, those concentrations align very well with the presence in those tissues of enzymes that require vitamin C to work. And those enzymes are copper and iron containing enzymes that need vitamin C to maintain activity. And if we plot now what we know about those enzyme activities and where vitamin C is high in concentration, you'll see that the two things always coincide. In fact, they even coincide to within the cell and within the subcellular compartments. So we know that vitamin C in the cell is also shuttled between compartments. Seems that it follows these enzymes around. And so the first one discovered was the collagen hydroxylase, and everyone in this room will have been seen in a chemist, creams with vitamin C added because it's good for your skin. So that little bit of knowledge that's translated now into a cream product is due to this activity here. So this enzyme was discovered in the 1960s, and immediately it was thought that's the reason that you get scurvy, is because that enzyme doesn't work and your collagen falls apart. Well, turns out that enzyme has a whole lot of cousins. And so we now know that there's probably about 70 enzymes in this category, and they do all kinds of things. So the copper enzymes in particular are responsible for hormone synthesis, and so we know the copper enzymes associated with adrenaline synthesis and with some of the brain hormones like serotonin as well. So there's a lot of hormone synthesis that's dependent on vitamin C. There's the synthesis of carnitine, which provides our ability to metabolize a high energy source. The HIF hydroxylases that we'll be talking about, which regulate the hypoxic response, so our stress responses to low oxygen. And in the last 10 to 8 years, there's maybe 15 to 20 of these enzymes, DNA and histone demethylases, which are enzymes that regulate the epigenetic processes. Now, epigenetics, for those of you who was pulled up yesterday for using this word, is the process within all our cells that tells each cell which genes it should read. So all our cells have the same genes, but different ones are read in each different cell. And epigenetics is that process, and it's very important, can be very important for early development and can be dysregulated in cancer. So this is how vitamin C works in that role. And so the enzymes undergo a cycle as part of their action. They also need, in addition to vitamin C, they need oxygen, and they need 2-oxyglutarate, which is a metabolite from our energy metabolism. So these enzymes are sensors for low oxygen and for energy metabolism, and also they will vary, their activity varies depending on the availability of vitamin C. And so vitamin C, which here called ascorbate, helps the enzyme cycle and keep active. and in that process can be consumed. So I'm going to give you 3 examples because the question we have, okay, well, you know you need vitamin C for that, but how much do you need? And does it matter if you're optimal or suboptimal? Like, do you just need enough to stave off scurvy, which we know is very little vitamin C, or does it make a difference to all of these enzyme activities if the vitamin C is optimal? So I'm going to give you 3 quick examples. The first one is the regulation of the hypoxic response. So in this response, this is a stress response that is ready to go in all of our cells all of the time. It needs an off switch. And the off switch are these enzymes that need vitamin C to work. So under normal circumstances, the best circumstances, this switch is off. We need it to go on only when it needs to. And so these enzymes here with the star, these are the ones that need vitamin C to work. They also are oxygen sensors. So when they don't work, this becomes active. So HIF1 controls many genes, including the genes that regulate glycolysis, cell growth and death. There's hundreds of genes that get switched on by this factor. So it's very important to control it in terms of controlling cell fate. And so we've done a lot of research into this, particularly around the cancer story, and what we see, so the question being, does it matter how much vitamin C your cells have got in terms of regulating this HIF1 response? Now, this is a little bit complicated, this picture, but essentially in these cells, and these are mouse cancer cells, when we've taken them and we've loaded them, either they have no vitamin C, and that's the black line here. So under the conditions of no vitamin C, as you remove oxygen, your HIF1 response comes on really quickly. So you can be at 5% oxygen, which is actually normal tissue oxygen levels, and your HIF1's already on. If you give those cells a small amount of vitamin C, so these conditions are for loading cells in vitro, then you get a muted HIF1 response. It will still come on when it needs to, when the oxygen is completely run out, or almost completely run out. If you give them optimal vitamin C, then you find your HIF1's not coming on until it absolutely needs to. And so in these cells, it makes a difference whether you have a little or a lot. And we think this is probably pertinent to much of our discussion today. Second example is carnitine synthesis. So carnitine is a molecule that requires vitamin C to be made, and it has, in 2 steps, it needs vitamin C for this step and for this step. And so it's a molecule that we need. It helps transport fat into the mitochondria. So it will help your cells maintain a normal oxidative energy production. So, so this is what it does. So it transports fats into the mitochondria. And here's a paper that I built from 2006. It's a little bit complicated. I'll just talk you through it quickly. So these are young adults who were supplemented with vitamin C or not, and they looked at their ability to metabolize fat instead of protein for energy, depending on their vitamin C status. And so you can see here they had a placebo group. So at 4 weeks, the beginning of the intervention, no difference between the control and placebo. This is their blood levels of vitamin C. So they took their blood levels from here, the black bars, just look at the black bars, that's the average from here to here. When the fat energy used at the beginning was not different, but when you upped their vitamin C level, they were able to use a lot more fat for energy when their vitamin C levels were optimal. Their protein went the other way around. So where these people with low vitamin C had compromised ability to metabolize fats for energy. Once you boosted them, that was restored, and their use of protein for energy went down correspondingly. So once again, makes a difference whether or not your levels are high or low. So not just any, but difference between high and low. The third example is something that's quite recent, and it's these histone, or these demethylases, the epigenetic enzymes. Now these are enzymes that also need vitamin C as a cofactor. So the epigenetics either puts marks on the DNA or on histones, removes those marks, or reads those marks. These enzymes belong to this category here. They remove the things that block gene expression. So that detail doesn't matter so much. At this point, there are up to 12 of these enzymes and 3 of these known. So there's now at least 15 of these enzymes that are working to control the basic development and expression in all of our cells. So very exciting discoveries in the last couple of years around these, and in particular with this study here, which was published in 2017, at the end of 2017. So what these people found, so these enzymes are important kind of for early development of cells, so for differentiation, so for stem cell differentiation. And one of those places where that happens is in our bone marrow. And so in your bone marrow, you've got stem cells that are differentiating into all the different blood types. So what these people found was that those early stem cells, which are here in pink, had very high levels of vitamin C, 20 times higher than the other cells, which we already know have high levels. And they thought this subtype of cell, all of that vitamin C, is once again a case of the vitamin C tracking those enzymes where they're needed to be active, because it's at this stage, the early development stage, that epigenetic writing is happening in those cells. So interestingly, these cells had hugely high levels of vitamin C. What they then did was they took the Gulo mouse, which is a vitamin C deficient mouse model, and they made it vitamin C deficient. And so they didn't do anything else, they just made it less vitamin C, and what they discovered was that the number of these cells increased in the bone marrow. So these mice that were vitamin C deficient started looking like pre-leukemic mice. So those cells were then they had decreased vitamin C status, and they changed their properties. So without any genetic mutation or anything that made it look like a cancer, they were starting to develop a pre-leukemic state. And they also had these stem cells in the spleen. So this is a little bit is not enough. You need optimal levels to maintain the levels in the blood. So does this, or inside our cells. So how does this apply to all of us? So this is what we see in the general population. Some European populations trend a little bit further towards saturation levels. But this is a New Zealand 50-year-old average population in 2010. So just taken from the electoral roll. And you can see that plasma vitamin C status varies across the population. So with 50 micromolar saturation levels being up here, not many people are walking around with optimal vitamin C status. So you'd want to be, up in this category here. So there's quite a lot of our population who are lower to start with. And so that's our public health message, probably, is it probably does matter how much vitamin C you take in. That, what we will be hearing about today too, is how much vitamin C do you need under different circumstances. And one thing that we know is that when you're sick, your vitamin C consumption increases. And so if we were animals, we would up our synthesis of vitamin C, and animals can increase the liver synthesis up to 100-fold to compensate for the increased turnover. we need to resupply. We need to maintain the supply. And so this is one of the things going to be a theme for today. But there are, this has been documented that, so these are deficiency, how many people in all these various states are actually deficient. And so you can see infectious diseases really high, cancers also, but unless people are actively maintaining their vitamin C status, their levels will deplete. So just quickly, does that matter? So what happens? What happens with those different vitamin C status? So we've actually measured how well the vitamin C transports into cells using a very clever tool just up the road and a very clever mathematician who can work all this out. And from that, we've been able to model this. So what you can see here, so this is like a snapshot through a blood vessel and into the tissues. And with this being the blood vessel and with this being distance in the tissues. The black line is the distance you would normally have between blood vessels and normal tissues. So if you're at deficiency level with vitamin C, you will just get very low levels of vitamin C just adjacent to the blood vessel. Doesn't go any further. If you have 50 micromolar, so that level that your doctor would say you're fine, then actually we find the vitamin C gets higher just around the blood vessel, but it doesn't get to the next blood vessel. So you will still have tissues that are low or deficient. You need 100 micromolar for that red line to move beyond the black line. And if you want it to go, if that distance becomes greater, then you need above the 100 micromolar for the vitamin C to diffuse everywhere through the tissues. That's probably important in cancer, where the blood vessels are really disorganised, and that intervessel distance is quite big. So we are thrilled with this data, and thank you to Kevin Hicks from the University of Auckland for his help with that. So I'm going to stop there. Hopefully I've seeded some of the future discussion for the day. I just want to thank some of my wonderful colleagues, Gabby Dax, who's here in the audience, Anitra, of course, Juliet Puller, who's also here. These students who have worked with us on the cancer projects, Helen Moran from the Tissue Bank, and a whole bunch of clinicians who are happy and willing to work with us and put up with us, and also our various funders. So thank you.